Welcome. In this video, I'm going to begin a new chapter, sexual reproduction in flowering plants. Now, whatever the concepts we have learned in the earlier chapter, that is reproduction in organisms, so with the help of those basics, we will try to understand this chapter, sexual reproduction in flowering plants, in detail. Now, if you look at the title, sexual reproduction in flowering plants, we know that out of all the plants in the entire plant kingdom, the only plants that produce flowers are angiospermic plants. So it means that we are going to study sexual reproduction in angiospermic plants, and all the flowering plants show sexual reproduction. So all the flowering plants, they show the sexual reproduction, and we know that how intimate relationship we have with the flowers, right? And human beings are using these flowers for various purposes, to convey happiness, grief, and for various other purposes, right? And here in the last chapter, as we studied, the sexual reproduction involves three main stages. One, it is pre-fertilization, then fertilization, and post-fertilization. So here also we will study the same thing. First, we will see about the pre-fertilization events, how they take place in these flowering plants, that is angiospermic plants, right? Now let's see about the pre-fertilization, right? So that includes the structure and events. So the pre-fertilization, structure, and events. So now when we see in case of the flowers in angiospermic plants, right? So there will be several hormonal and structural changes that actually take place to initiate the production of flowers. Now the flowers are actually modified. So flowers are modified shoots. So the shoot gets modified to produce these flowers, and for this there will be several hormonal and structural changes take place, and these hormonal and structural changes will lead to differentiation and further development of floral primordium. So further development of floral primordium. So here the plant, when it requires to produce these flowers, there will be hormonal and structural changes that take place that will help the shoot to get modified and produce the flowers. Of course, in some cases the flowers are clustered to produce inflorescences, that I think we have already studied in the first year about the inflorescences, right? So now whatever that hormonal and structural changes which will lead to the differentiation and further development of this floral primordium. So the floral primordium is the flower at its early stage of development. So the flower at its early stage of development, so that's what we call it, the floral primordium. So in the floral primordium, the parts of the flower are not yet differentiated. So there are nothing like sepals, petals, androecium, gynoecium, which are not seen clearly in case of this floral primordium. So later, as this floral primordium develops and undergoes differentiation, we will be able to see those sepals, petals, androecium, gynoecium in detail. So now let's look at that structure, that is the flower, in detail, and see the parts of the flower. First, parts of the flower. So when we see these parts of the flower, these I'm going to divide into two categories mainly. One I call non-essential floral parts and another one called essential floral parts. So I categorized the parts of the flower into two categories. One, it is non-essential floral parts. It means these are the parts of the flower which will not involve in the reproduction. So they do not involve in reproduction. So whereas essential floral parts are the one which involve in the sexual reproduction. So they are involved in the sexual reproduction. So now let's look at what are those non-essential floral parts. The non-essential floral parts are two. So there are sepals or calyx, then corolla, right, or the petals. So petals or corolla. So these two are non-essential floral parts which will not be involved in the reproduction process. Whereas the essential floral parts are again two. So that is androecium and the gynoecium. So this is the male reproductive organ. So androecium is the male reproductive organ, whereas the gynoecium is female reproductive organ, right? And the members of this androecium are called stamens. So members of androecium, so they are called stamens, whereas the members of gynoecium, so they are called the pistils. So later I'll discuss about the structure of those stamens and pistil. So now all these four parts, right, they are arranged in the four whorls on the swollen part of the flower, that is thalamus, right? So when we see the flower, right, so the base of the flower is swollen. So this part, it is called the thalamus, also called receptacle, right? And located on this thalamus we find four whorls. So each whorl is occupied by, right, these parts of the flower. So one whorl it is occupied by sepals, another whorl occupied by petals, and then androecium, and then gynoecium, right? So there are four whorls on the thalamus, four whorls on thalamus. So I can say that each whorl is a circular structure, so that is occupied by the members of the flower. So that may be the sepals, petals, and so, right? So if we look at that four whorls on this swollen part, that is thalamus, so these are four whorls, right? So we see this is the first whorl, right? That is the outermost whorl, right? And below that we have is the second whorl, and this is the third whorl, and this is the innermost whorl, which is the fourth whorl. Now the first whorl, of course, I'll just mention here, is the innermost whorl. So the outermost whorl is occupied by the sepals or the calyx. So is occupied by sepals or calyx, whereas the second whorl, this is occupied by the corolla or the petals, right? So these petals may be colored, right, in some cases, and will produce scent and so on. So that we shall see in our coming classes. Then the third whorl, it consists of androecium. So that is stamens are present in this whorl, that is the third whorl. Whereas the innermost whorl is the one that consists of the female reproductive organ, gynoecium. So that consists of one or more pistils. Right. So this is about the four whorls of the flower. The first whorl, the second whorl, the third whorl, and the fourth whorl, which is the innermost whorl that consists of the female reproductive organ. So the first two whorls are non-essential, occupied by non-essential floral parts, whereas the the innermost two whorls, they are occupied by essential floral parts. So now let's see that how it looks like. So this is the thalamus. On the thalamus, the outermost whorl, so that is occupied by, so these are sepals, and below that you find the petals. So this is a petal and sepal, and then here you will find, so you can see this is the outermost whorl that is occupied by the sepals, right? And next to that will be the second whorl occupied by the petals or the corolla, right? And then the third one is occupied by the stamens, and at the center you find the presence of the gynoecium or the pistil. So this is the stamen, and this one is the pistil, and this is thalamus or receptacle, and this is the stalk of the flower, that's called the pedicel, right? So this is how we see the flower in longitudinal section. So that is LS of flower, right? I hope this part is clear. So now let's see. So as I'm not going to discuss about the sepals and petals, so we will straight away move on to the essential floral parts, so which involve in the sexual reproduction. So I'm going to discuss about the androecium first. So we know the members of this androecium, so they are called the stamens. And when we see the stamen, of course, I just say the stamen is the microsporophyll. So it can be comparable to the microsporophyll. And these stamens will be having two parts. One, it is anther, and another structure is called the filament. So this filament is a long and slender stalk. It's a long and slender stalk on which attached is the anther, right? So this anther is a bilobed structure. Anther is a bilobed structure. So I can compare this anther with the Microsporangium. So anther, microsporangium, and inside this anther will be the development of pollen grains. So that pollen grain I will compare to the male gametophyte. So you need to remember these are synonyms. So sometimes instead of using the pollen grain, they may use the word the male gametophyte. So if they use that word male gametophyte, then it is actually the pollen grain. And the microsporangium, which is the part of this anther, and the stamen in fact is the microsporophyll. Now when we see here the anthers, so this is a bilobed structure, and you will find here this is the filament which is attached. Of course, the way the filament or the stalk attached to the anther varies. Okay, so in this case I say the filament is attached at the base of this anther, so I can use a word like base fixed, and there are several types of this that we shall see it later. Now, so this is the anther, which is a bilobed structure. So on this is anther, right? So two parts of the stamen, that is anther and filament. And the proximal end of the filament, so this is the proximal part of the filament. So this proximal part of the filament, the proximal part of the filament is attached to thalamus, or may be seen attached to petal. So in some flowers you may see it is present on the thalamus, or it may be seen fused with the petals. So here in this case I'm just drawing to show that it is present on the thalamus, the proximal part. So this is the proximal part of the filament, and when we see the distal part of the filament, it is seen attached to the anther, right? So next the number and length of the stamens. So here the when we see in this case the filament, in some the filaments may be much longer, in some the filaments may be shorter, and the number of stamens may differ from species to species. So in one species there may be 10, in another species it may be 12, and so on. So different species will have different number, right? And even the length of that stamen will be two species. So in one species there may be 10, in another species it may be 12, and so on. So different species will have different number, right? And even the length of that stamen will be changed, or it's different. So I say the number and length of stamens in different species is variable. So is variable. So now let's see the structure of anther in detail. Right. Now this anther in a typical angiospermic plant, in a typical angiospermic plant, so it is bilobed, so it has two lobes, and it is dithecous. So here it has, each lobe has two theca, each lobe has two theca. So here the word theca, which means cover, right? So it's a cover. So here each lobe shows two covers, which we call the theca here. Now let's take the transverse section of anther. So here we have seen this the anther, and I'm going to take the TS, the transverse section of the anther. So when we have taken the transverse section of the anther, so it may be seen somewhat like this. So you can see this is the structure of the anther. Right, in the transverse section. Now in this case you can see that there are two lobes, So this is one lobe, this is another lobe, and each lobe, as told here, is covered by, okay, two theca. So you can see here over, this is a theca covering here, this is another theca. So I can say that it is dithecous. So this condition, so as there are two theca, the condition is dithecous. So as I already told you that anther in a typical angiospermic plant is bilobed and dithecous, right? And here, of course, this is the connective, so which consists of, so it contains vascular bundles. So that will be containing the xylem and phloem, which will help in transportation of minerals and nutrients to the anther or the tissues of the anther. Now you can see here there are four microsporangia. So here each one is a microsporangium. So microsporangium. So microsporangium is the structure. Inside this will be the development of pollen grains. So microsporangium is a part of the anther inside which development of pollen grains take place. So inside which the development of pollen grains will be taking place. So that's you will find a total 4 microsporangia. So there are 4 microsporangia present. So hence I call this condition as tetrasporangiate. So where the tetra, which means 4, so 4 sporangia are present, right? So you can see in each lobe there are 2 microsporangia. So each lobe has 2 microsporangia. So in this case you can see that the 4 microsporangia present at the 4 corners. So I can say this anther has 4-sided, that's a tetragonal in shape. You can see that tetragonal in shape, right? And longitudinally you will find along the, that is along the length of the anther, you will find something called the line of dehiscence. So the line of dehiscence means it is the point where, okay, it splits open so as to release the pollen grains, right? So from this is the point from where the pollen grains, so you can see that one. In this case, this is the line of dehiscence. The line of dehiscence. I can use another word called stomium. The line of dehiscence, right? So these microsporangia are going to further develop into pollen sacs. So microsporangium, so as the anther develops, so they are going to become into the pollen sac, so inside which the development of the pollen grains will be taking place. So of course here at the center of this microsporangium, each microsporangium, we find sporogenous tissue. So it consists of sporogenous tissue. So this is the tissue which will be undergoing meiosis and later develops into the pollen grain. So what I say, this is the sporogenous tissue. I'm going to discuss again with that with the sporogenous tissue how the meiosis process takes place and results in the formation of the pollen grains. So in the process called microsporogenesis, right? So this is how the transverse section of the anther is seen. Now let us see in detail right about one microsporangium. So what is the structure of microsporangium? So let's see structure of microsporangium, right? So here just I'll draw the same part, the anther, okay, transverse sections. Okay, so these are the four pollen sacs or the microsporangia. So I'm going to just magnify this part and show you that what are all the structures that we see along with this, along with the microsporangium. Now, a typical microsporangium, a typical microsporangium appears almost circular in shape. Appears almost circular in shape. So you can see which I have drawn here, almost it's circular in shape. And the wall of the microsporangium is surrounded by four wall layers. So the microsporangium wall has four layers. So it has four layers or four wall layers. So here this is the thickness of that wall. So that I call the wall of the microsporangium, and it has total four layers. So they are: one, epidermis, and then the second layer will be the endothecium, and the third one will be the middle layers, and the fourth one is the tapetum. So these are the four layers. So this epidermis is the outermost layer, whereas the tapetum is the innermost layer. So outermost is the epidermis and innermost is the tapetal layer, right? And the first three layers, that is epidermis, endothecium and middle layers, these will give protection. So they give protection and also help in dehiscence. So dehiscence, which means the split opening of the anther and releasing the pollen grains, is what we call the dehiscence, right? And that will take place at the point what I mentioned, which already the line of dehiscence, that's the stomium, right? So this epidermis, endothecium and the middle layers will give protection and help in dehiscence, whereas the tapetum will, okay, or it helps in giving nourishment, means nutrition, okay, to the developing pollen grains, to the developing pollen grains. So right, so the tapetum, which is the innermost layer, and it will be the one which will be in contact with the developing pollen grains, and this will help in giving nutrition to those developing pollen grains. So now let's look at this structure diagrammatically. So as I have mentioned, the outer layer epidermis, below that endothecium, the middle layers, and tapetum. So these three layers will have the protection and help in dehiscence. That's the function of those three layers, whereas this inner one, that is the tapetum, will be helping in nourishment to the developing pollen grain. So now let's look at the diagram. So here this is the outermost layer, that is epidermis, and below the epidermis we find endothecium. So that's an endothecium layer, and then below the endothelium layer we find the middle layers. So these middle layers may be 1, 2, 3 layered. And the innermost layer is the tapetum. So this is the outermost layer, epidermis, and below that we have is endothelium, and these are the middle layers, and this one is the innermost layer, that is the tapetum. So here whatever you find, all this is the sporogenous tissue. So this is going to undergo the process of meiosis, and that results in the formation of the pollen grains, right? So this is the sporogenous tissue, and this one is the pollen sac, or I can say the microsporangium, right? I hope that's the part is clear. Now one more point in this case about the tapetum. So this tapetum, it has dense cytoplasm. So it means its cytoplasm is thick due to presence of various substances including the nutrients. Because of that, it is having dense cytoplasm, and it also has more than one nucleus. So it has more than one nucleus. So I can say it may be a binucleate condition, or it may even be a multi-nucleate condition. Binucleate, where you find two nuclei. Multinucleate, or more than that you will find, or many nuclei also seen in case of the tapetal cells. So the tapetal cells have dense cytoplasm and usually have more than one nucleus, where I'm mentioning it as a binucleate or multinucleate condition. So this binucleate or multinucleate condition can arise due to endomitosis. So it can arise due to endomitosis. So endomitosis, where the nuclear divisions take place, but it will be not followed by the cytokinesis. Because of that, these cells, that is the tapetal cells, will be having more than one nucleus, right? So you can see more than one nucleus. So the tapetal cells will be having more than one nucleus. That is because of a condition called endomitosis, where the karyokinesis is not followed by the cytokinesis. Because of that, the condition like the binucleate or multinucleate condition arises in the tapetal cells. Right, so with this I will end today's session. I shall continue this one in the next video. Thank you.